Hey guys, welcome back to our third episode in our genetics series. If you haven't seen episodes 1 and 2, please definitely check them out before watching this video. In today's video, we're going to be looking at something called Punnet Squares, which help us work out what kind of offspring we can get if we know what the parents are. Let's get to it. So guys, in previous episodes we looked at what we meant by alleles. We looked at inheritance from sexual reproduction, and we looked at some of the key terms that we need to know for simple genetics. Today we're going to go on a little bit further into inheritance and show you a way that we can easily calculate the possible outcome or the possible offsprings from two different parents based on the knowledge of the genotype and phenotype of those parents. Now, if you don't remember what any of these terms were, check our previous episodes on the key terms and make sure you watch those to have a bit better understanding about what we're going to talk about today. So we're going to look at something called Punnet Squares. And these are a little sort of almost diagram which we can use to look at two different parents, two different organisms, and predict the possibility of what they can produce in terms of offspring. Now we're going to start with the same example we've used before, where we had a female who had two copies of the blue-eyed gene, and we gave them the letters little b, and we had a male who had two copies of the brown-eyed gene, and we gave them the letters big B, big B. So now we know what the genotype for the parents are. We can use a Punnet square to predict the possible offspring. So to draw a Punnet square is very simple. What we do is we draw a 6x6 grid, or 2 lines down and then 2 lines across like this, and then we can use this to help us work out what the possibility of offsprings are going to be from these two parents. So let's take the first parent, for example. Now it doesn't matter which parent goes where, but for this sake we're going to put the female down the side. So we're going to take her first allele, which is little b, and we're going to put that there, and we're going to take her second allele, which is another little b, and put it there. Now the male is going to go along the top for this example. Big B will go up here, which is the first allele, and the second allele is big B. Now what we're going to do is we're going to fill out each of these 4 squares. So for each for this pairing is essentially we can have 4 possible offspring, and we can predict that using this Punnet square. So for this offspring, it will be capital B from up top and little b from here. For this offspring, again, you bring down the top, capital B, and little b from this column, little b. Same here again, comes down, big B, comes across, little b, and then comes down again, capital B, and comes across here, little b. Now you can see from this Punnet square, if you had these two parents, 100% of the offspring, all of the offspring are going to have the genotype, capital B, little b, and the phenotype, brown eyes. And again guys, if you don't understand where we got brown eyes from, watch the previous episode where we talk about dominant and recessive, and we use this example. Now we also know they're going to be capital B, little b, and the term we used for that was heterozygous. So all the offspring in this example are going to be 100% heterozygous, capital B, little b, and they're all going to have the phenotype brown eyes. So let's take a look at a slightly different example where we can look at the Punnet square for two different parents. So we can see in this example, we have the same mother and she is homozygous. So she's got two of the same genes, two of the same alleles, little b, little b. So she's a blue-eyed mother with little b as her genotype, but we've changed the male here. We've changed it, instead of it being a homozygous brown eye, so two capital B's, it's now actually a heterozygous male. So it's got one copy of the brown-eyed gene and one copy of the blue-eyed gene. But remember guys, because brown is dominant, this male, right, will still have brown eyes. We'll still see brown eyes even though it's carrying this blue-eyed gene, because the dominant gene is masking that. So we can look again at what we can get out of this pairing. So we will again draw a Punnet square, 6x6 grid. There we go. And we'll do the same as we did last time. So we'll take the mother's genotypes, or the mother's alleles, so little b, and the father's alleles, big B, little b. Now remember, the reason that we do this is that only one of each parent, when a offspring is made, it can only pass on one of its two alleles. It cannot pass on both. That's why we break down the possible gametes, or the possible sex cells. In this case, for the male, the sperm cells could either be carrying just the big B or just the little b. With the mother's, or the female in this case, her egg cell could either have a small little b or a small little b, because she has two copies. So it could either be carrying this version of the little b or this copy of the little b. So we'll do the same thing again. If we bring that down here, we have capital B, and from there little b, come down, capital B and across, little b, come down, little b and across, little b, and then come down, little b and across, little b. And you can see this time we have a slightly different result. What we have is 50% of the offspring are going to be heterozygous big B, little b. So 50% will be big B, little b, heterozygous, and have the phenotype, the visual characteristic of brown eyes. With the other 50%, however, you'll notice that we have half of the possible offspring. Remember, this doesn't mean that exactly half of the offspring will be this and half the offspring will be that. We're saying for every time an egg is fertilized, you have a 50% chance of it being capital B, little b heterozygous, which is brown eyes, or you have 50% chance of it being little b, which is homozygous. And if you remember from our previous video on dominant and recessive, where there are two recessive genes here, together you will see the visual manifestation of this recessive trait, which would be blue eyes. And there we have a different scenario because we have a different male. So let's look at one final example where if we had two heterozygous parents. So here we have a female and a male which are both heterozygous, meaning they each carry one copy or two different copies of the same allele. In this case, each one carries an allele for the brown-eyed gene and the other one for a blue-eyed gene. So visually, these would be two brown-eyed organisms. If we're talking about people, this would be two brown-eyed parents, okay? Because with the dominant gene present there, it will be masking that recessive gene. So here we'd have brown eyes and here we'd also have brown eyes, but they are carrying the gene for recessive brown eyes, okay? Blue eyes, sorry. So let's have a look at that again. So we'll draw the Punnet square, and we'll take the genes from the mother, capital B, little b, and the genes from the father, capital B, little b, and we'll repeat the same process once again. So coming down, capital B and across, capital B coming down, capital B coming across, little b and down, little b across, big B and down, little b and across, big B. And once again, you can see a different result. Now there is something key to point out here. Where we see these two genes, or these two combinations here for the offspring, we see this is capital B little b and this is little b capital B. They are in fact the same thing, just written in reverse. It just means that both of these offspring, so these two here, would be heterozygous offspring. Now we can look at what percentage it is by looking at it's 2 out of 4, which is 50%. So 50% from this breeding, you have 50% of the offspring would be capital B, little b. Remember guys, you can write this the other way around. However, we generally like to write the capital first. So capital B, little b, and it is heterozygous heterozygous, and the phenotype, the visual manifestation, again, because the dominant will mask the recessive, will be brown eyes. If we look at the remaining, we have this here on its own with 2 capital B's. So out of a percentage, it's 1 out of 4, or 25%. So 25% chance that the offspring will be capital B, capital B. And remember, when there's 2 of the same, we say it's homozygous. So 25% chance that the offspring will be homozygous capital B, and remember capital B stands for brown eyes, so here we would have brown eyes. And finally, you would have this one here, where we're looking at 2 little b's, again 1 out of 4, so 25% chance that it would be little b, again 2 of the same, which means homozygous, and therefore blue eyes. Key to note here, however, you've actually got a 75% chance, also so 50 plus 25, you have a 75% chance that the offspring will have brown eyes. However, 25% of all offspring will be brown eyes but homozygous, and half, 50% of all possible offspring will be brown eyes also, but they will be heterozygous. But there would be no visual way to tell these 2 offspring apart. There would be no way to determine whether a brown-eyed offspring was heterozygous or homozygous until it grew up and potentially bred with, let's say, in the case of homozygous, if it bred to a homozygous blue-eyed person in this case, someone with blue eyes is definitely homozygous because of the way recessive genes work. If you then had 50% of the offsprings coming out with blue eyes, you would know that the brown-eyed parent was heterozygous. Or if you had all offspring came out brown-eyed, then there is a chance that the parent was actually homozygous, which means all of the offspring would carry at least 1 copy of this dominant gene, going back to our original scenario with the homozygous, 2 different homozygous parents. Right guys, I hope things are all coming together now with that Punnet square and the 2 previous videos. Hopefully from the next episode, episode 4, we'll be looking at how these things apply to ball pythons, guys, and how we can take what we've learned so far and really apply it to breeding ball pythons and genetics behind them. Don't forget to hit the like and subscribe button, guys. Your support means the world to us. Leave a comment in the comment section down below, and until the next episode, we'll see you then. Bye.